Welcome back to Dissect the Question. A graduate student is studying two types of muscle tissue to better understand the mechanisms of muscle contraction. He examines both smooth and striated muscle tissues under laboratory conditions, inducing contraction in both samples. Which of the following contractile mechanisms is required in striated muscle only? Let's see, striated muscle. So that's the key, striated muscle. So striated muscle is both voluntary skeletal muscle, and we have striations also in the involuntary cardiac muscle. So let's see, it stands out immediately to me that calcium troponin C binding might be the answer. But we can't stop there. Even if we think we know the answer, we have to look at the other choices because often there are two correct choices and we're always required to select the best one. So if we look at calcium troponin binding, we know that troponin, here's a sarcomere in striated muscle, and these are Z lines, and actin is attached to the Z lines. Those are the thin filaments. This is the contractile mechanism according to the sliding filament theory. And here we have myosin, the thick filament. And when calcium enters, after there's a depolarization from the motor neuron releasing acetylcholine to the muscle, we call that excitation-contraction coupling. And when we have a depolarization along the Z lines, there are T-tubules, and calcium enters. Troponin is one of the subunits, specifically troponin C we're talking about here. Troponin C is one of the subunits of actin, isn't it? And so our troponin is sitting here on top of the actin filament. This is actin. And when calcium enters, it allows the troponin to interact with the myosin and producing this interaction leads to cross-bridging. The cross-bridging produces active tension. Active tension. That's the tension we need for purposeful movement. When we're trying to actually lift a load, we call it purposeful movement. So we need that cross-bridging to occur, and according to the sliding filament theory, the Z lines will come closer together. This sarcomere will shorten, and that produces the active tension. So clearly we have calcium troponin C binding going on in the striated skeletal muscle. Now what about E? It also jumps out at me that there's a presence of intracellular calcium here, and is there a presence of intracellular calcium only in striated muscle, only in cardiac and skeletal muscle? No, there isn't. We know that intracellular calcium is the main signaler for contractile force in all muscle, including smooth muscle. Smooth is another type of involuntary muscle, in addition to the striated cardiac muscle. Now, what about the other choices? So we can eliminate E because it's present in all, and here, this question is asking striated muscle only, meaning skeletal and cardiac. Cyclic GMP is an intracellular signaler, most important in smooth muscle, not striated muscle. We know that nitric oxide, NO, is associated with this mechanism where nitric oxide causes smooth muscle to relax, and in the blood vessels, it produces what we call vasodilation to increase blood flow. So, that's increasing blood flow to the tissues. So, cyclic GMP does not play a big role in the contractile mechanism. Although there might be some cyclic GMP present in striated muscle, it's not part of the contractile mechanism. And nitric oxide is more important in smooth muscle contractile mechanisms. Myosin light chain phosphorylation and myosin light chain phosphatase activation are both smooth muscle activities. So we don't have significant amount of myosin light chain phosphorylation by, remember the enzyme, myosin light chain kinase, and phosphatase, myosin light chain phosphatase. Myosin light chain kinase initiates contraction of smooth muscle. Myosin light chain phosphatase initiates relaxation of smooth muscle. So we can eliminate B, C, and D, and it seems like A has to be the only appropriate and best answer.